hear us as well, or at least me. Okay, so I think I can start. So, yeah, hello. Oh, is there still an echo? Okay, perfect. So let's go. My name is Raquel, and today I have the pleasure to introduce Dr. Kevin Tracey in our Brain Body Interaction virtual seminar series. Just as a reminder, these seminars happen every Monday, and we are bringing together scientists which are interested in the interface between the nervous system, organ physiology, immunology, reproduction, aging, cancer. So stay tuned for updates in our website and also in Worldwide Neuro Seminars. And before introducing our wonderful speaker, let me remind you that you can ask any question during the talk using this Ask a Question button. And also, if you want to meet the speaker after the talk, we are going to have a Zoom Q&A session that I will be placing the link here in the chat in a little bit. And so, yeah, now for real, I'm eager to introduce Dr. Kevin Tracey. I'm going to briefly summarize his really impressive career so we have some time for his talk. Dr. Tracey is president and CEO at the Feinstein Institutes for Medical Research, professor of molecular medicine and neurosurgery at the Donald and Barbara Zucker School of Medicine at Hofstra Northwell, and also executive vice president at Northwell Health. He received his bachelor's degree in chemistry from Boston College and his MD from Boston University. He trained in neurosurgery at the New York Hospital Cornell University Medical Center and was a guest investigator at the Rockefeller University before moving to the Feinstein Institute. There, he directs the Laboratory of Biomedical Science. He is a leader in the scientific fields of inflammation, bioelectronic medicine, represented by his more than 120 United States patents and more than 400 scientific publications. I think I'm late already for this. He also co-founded the Global Sepsis Alliance, which is a non-profit organization supporting the efforts of more than 1 million sepsis caregivers in over 70 countries. So the major focus of Dr. Tracey's laboratory is the molecular basis of inflammation and the mechanisms by which neurons control the immune system. So today he will be talking about reflex regulation of innate immunity. Thank you so much, Kevin, for being here with us, and the stage is all yours.
Thank you very much, Raquel. It's really wonderful to be here. Can you hear me okay?
Yes, everything is perfect. We
Great.
just cannot see your presentation yet.
We shall try to share the screen. The circle's going round for sharing the screen. How's that, Raquel?
It is fantastic.
Can you see my
we
slides?
can hear you. We can see the slides. Thank you.
We can hear you. We can see the slides. Terrific. So thank you very much for your kind words. Thank you, Raquel, and to all the organizers for the invitation to be here. I would rather be with all of you in person wherever you are, but this is great to be able to share some thoughts on the intersection of the nervous system and immune system. As you heard, my particular interest with my colleagues here in the laboratory at the Feinstein Institute is in studying the neurological basis for the control of inflammation or innate immunity by reflexes. But before we get to that, I would like to create a little context, particularly for the younger people on this call who are thinking about various choices in their career paths in neuroscience and immunology, and just to frame for a few minutes the importance of where we are today in these kinds of questions. So let's begin with a little quiz. So I can't see you, but you can shout out answers or type in the chat box. In the history of the modern human race, that is for approximately the past 50,000 years, when humans really began speaking, there have been 110 billion people born. And from then till now, approximately 102 billion of those people have died. So the question I have for you is, until almost 1900, what was the leading cause of death for all those 108 billion people who came before us and died? The answer is infection. Infection was the overwhelming, the singularly overwhelming commonest cause of death for modern human beings for the past 50,000 years. And when you look at this as a plot, as a curve, it's quite striking. Now, this is only the last 2,000 years, so you can put 48,000 years to the left of that. And what you see is that human life expectancy was almost completely unchanged until very modern times. But then something happened. Something happened that changed the curve in a dramatic way. And as students of science, I hope you all immediately guessed the answer was, well, what happened was the scientific revolution. And the scientific revolution produced the modern knowledge that provided the first scientific explanation for what was killing, the commonest cause of killing the human race. And you know, the theory that was displaced was actually the longest lasting theory in the history of medicine, the humoral theory of disease. So before the scientific revolution, all disease was believed to have been caused by an imbalance in bodily humors. In theories that were proposed by Hippocrates and then elucidated and captured further by the Roman physician Galen and his colleague Celsus, what these gentlemen proposed was that all disease could be explained when blood, black bile, yellow bile, or phlegm inside produced by the body became imbalanced. And therefore the treatment, they believed of all diseases was based on restoring the balance or the imbalance of these humors. And they did this by changing the diet, by changing the air, by bleeding to alleviate excesses, and the use of cathartics to induce diarrhea, nausea, and vomiting, all with the hope of balancing the humors. And you know, this theory was used to explain everything from gout to the Black Plague. So, you know, to put in context the plague we just lived through, in many ways there's no comparison. I mean, a typical plague that swept through the first 50,000 years of human survival would kill a significant fraction of the people living on Earth at the time. So just as one example, in the 14th century of the bubonic plague, which swept through Europe, it killed 1 out of every 3 people. And, you know, although the physicians at that time could characterize in some detail the signs and symptoms of these diseases, and they knew there were 2 different forms of the disease, a bubonic form, which would last for 5 to 14 days with large lymph nodes and a relatively high mortality, and another form, a septicemic form, which presented really as a fulminating case of septic shock, which killed 100% of its victims within 24 hours. You know, even today, it's not really clear what the source of these plagues was, or these pestilences, or these pandemics, although a likely cause of the 14th century plague was Yersinia pestis. Now, in the scientific revolution, people like Louis Pasteur established a replacement theory as a cause of disease, and that, of course, is the germ theory. I mean, Louis Pasteur, as a young boy, watched a neighbor of his in his neighborhood who'd been bitten by a rabid dog. He watched as the townsfolk held the little boy on the street and cauterized him with red-hot pieces of steel, cauterizing the dog bites in the hopes of preventing rabies. Of course, Louis Pasteur said he never forgot the screams of the young boy who he heard being treated in this way. And then one day, Louis Pasteur invented the vaccine for rabies. And he did this after working for many, many months, not knowing whether the pathogen he was passing from a dog to a rabbit brain, and then passing that pieces of rabbit brain and spinal cord from one rabbit to 20 or 25 additional rabbits, never knowing if that pathogen was a virus or a bacteria or what it was. He nonetheless was able to prove that pieces of the rabbit spinal cord, after being passaged 25 times, could protect 50 dogs from rabies. And then one day, on July 6, 1885, Louis Pasteur inoculated Joseph Meister. And he was the first human being to have sustained rabies and survived. So all human beings prior to the time of Joseph Meister who'd ever contracted rabies died. And Louis Pasteur, when asked what he wanted on his epitaph, said, "Tell them Joseph Meister lived." So from that, you can put your favorite heroes of science and medicine on this slide, but whether it's Edward Jenner or John Snow or Louis Pasteur or Professor Paul Ehrlich or even Alexander Fleming, these contributions of the scientific revolution drove largely the eradication of infection, all the way to today's modern vaccines, as most recently as last year. So if infection was the cause of death, and we've got that under pretty good control, then what is killing the human race today? Inflammation. Of the 58 million people who die on the planet Earth today, 40 million every year die of inflammation or inflammatory diseases. This is, of course, an abridged list, but these are the commonest causes of death of the 58 million people on Earth who die annually. These are the commonest causes of death. And all of them are either caused by, accelerated by, or enhanced by inflammatory mechanisms. Cancer, cardiovascular disease, dementia and other neurodegenerative disorders, diabetes, metabolic syndrome, obesity. The inflammatory contribution to the leading causes of death on the planet Earth today is the opportunity for future research. It's the opportunity for future healthcare. It's the opportunity to continue to make the contributions that the scientific revolution started onto human longevity. So today we're living in an era where inflammation is being treated. It's not being cured, but it's being treated. When I graduated from medical school in 1983, which, depending on your point of view, was really long time ago for some of you, or not too long ago for me, but anyways, inflammation, I was taught, is a condition in which part of the body becomes red, painful, and swollen because of injury or infection. Now, to be a little bit jaundiced about this, that explanation that I was taught in medical school is really not that different than the humeral theory of disease. It's really not significantly advanced. But from the time I was in medical school till now, there's been another revolution in understanding the molecular basis and the cellular basis of inflammation. And I call that revolution the cytokine theory of disease. Now, the cytokine theory of disease doesn't state that all cytokines are bad. And the germ theory of disease doesn't state that all germs are bad, frankly. The cytokine theory states that in some pathogenic conditions, cytokines are necessary and sufficient to cause the disease. And in those situations, patients can benefit by being treated with inhibitors of cytokines. Now, obviously, over the course of human evolution, cytokines evolved to maintain health. But what we now understand today is that an overproduction of cytokines is necessary and sufficient to cause inflammatory diseases ranging from rheumatoid arthritis to Crohn's disease to many, many other inflammatory conditions. And in those cases, the administration of cytokine inhibitors like anti-TNF and anti-IL-1 and anti-IL-6 can ameliorate and attenuate the disease. Now, all of these agents are clinically approved for use in humans, and they've been given to patients to their benefit of millions and millions of patients. So this has been a revolutionary concept in the idea that disease is caused by molecules made by the body, and that controlling those molecules is critical to maintaining health. Now, this occurred because in the last 30 years, there has been essentially a revolution in focusing scientific technology and knowledge and the tools of molecular biology on these fundamental questions of what controls and causes inflammation. There's also been, in the US and worldwide, a significant increase in dollars for research. And this has driven the cytokine theory of disease to now account for, directly or indirectly, as much as 20% of the global pharmaceutical industry. And I don't know, but I suspect that treating inflammation will begin to extend the lifespan curve again. And the question is, what would happen if it was possible to cure inflammation? If it was possible to cure inflammation, what would that mean for the longevity of the species or the lifespan of individual humans? I don't know, but it's certainly an interesting question, and it's certainly sufficient reason to pursue aggressively knowledge and understanding how to treat inflammation and what causes it, and to study mechanisms through the nervous system and others that might control inflammation. Now, one type of inflammation that can be devastating is Crohn's disease. Crohn's disease is an inflammatory bowel disease, and this patient, Kelly Owens, who I've come to know very well, by the time she was 30 years old, had spent 15 years debilitated by the disease, in and out of hospitals, in and out of wheelchairs. And the drugs I list here are the drugs that failed her. Some people say she failed the drugs. The patient doesn't fail the drugs. The drugs fail the patient. So after millions of dollars and multiple failed attempts to control her Crohn's disease, Kelly told me that most of the time, these drugs made her feel worse, not better. So with this in mind, some years ago, my colleagues and I developed a molecule that we hoped would be useful for Crohn's disease and other inflammatory conditions. And we named the molecule CNI 1493 after cytokine networks. 1493 for the year it was invented, 1492, but then we added one because we didn't want to compete with Christopher Columbus's designation of 1492. So we named it 1493. Now, this particular molecule is a tetravalent guanyl hydrosome because it has 4 groups on it that are arginine mimetics. And I won't go into the reasoning and all the story behind the development of this molecule, but what we showed was that this molecule was very good at inhibiting TNF, and that by blocking TNF production, TNF, of course, an early cytokine in the cytokine cascade or the cytokine storm, and by blocking TNF in animal models of inflammation and infection, by blocking TNF, CNI 1493 was a very good anti-inflammatory molecule and could reverse the signs and symptoms in numerous animal models of inflammation and infection, ranging from shock to sepsis to meningitis to arthritis and to colitis. And after many, many publications and years of work with dozens of colleagues and several companies, this molecule made its way into clinical trials for Crohn's disease, where we ultimately treated about 12 patients once a day in the early pilot trial, and saw that 8 of these patients had remissions. And these are, the green line is 4 milligrams per kilogram, and this is 25 milligrams per kilogram in the red line. It was a very small study, but there was a highly significant reduction in the Crohn's disease activity index. And on endoscopic biopsies, there was evidence of healing. So this is a severely inflamed colon of one patient, which healed significantly over a period of 4 weeks. So we were extremely excited and pleased with the progress in this development of this molecule as a cytokine inhibiting anti-inflammatory drug. And we pretty much thought we knew everything about it until one day we were in for the surprise of our lives. Because when we put the molecule into the brains of animals with a stroke, expecting to turn off the cytokines in the brain, the molecule in the brain also turned off the cytokines in the lung, the heart, the liver, the spleen, the kidneys, and the intestines. This was, frankly, this was inexplicable and unbelievable, and we really couldn't figure out how a small amount of this molecule in the brain could possibly have turned off the cytokine production in the body's organs. There wasn't enough 1493 in the brain to turn off all of the body's organs. When we studied this more carefully, Tom Bernick, a postdoc in the lab, showed that to the IC50 for inhibiting serum TNF in mice and rats when the drug was given IV was on the order of 0.1 to 0.3 milligrams per kilogram. But when he's put the same drug in the brain, he showed that the molecule was logarithmically more potent. So here, the IC50 for inhibiting serum TNF is between 0.1 and 1 nanogram per kilogram when the drug was put in the brain. So we racked our brains trying to figure out how this might possibly work, and wondered maybe the signals were being carried in the vagus nerve. And so Tom repeated those experiments, but this time he cut the vagus nerve. And now when he did that, the drug in the brain no longer blocked the production of TNF or other cytokines, IL-1 and IL-6, in the body's organs. So at this point in the story, we knew there was a signal that could arise in the brain, travel down the vagus nerve, and inhibit cytokines. But we can make no sense of how or why that would be. Then we came across a paper by Linda Watkins, in which Linda did an incredibly important experiment. She injected animals systemically, intraperitoneally with IL-1, and they developed a fever. And this was known. But what was not known, and Linda discovered, is when she cut the vagus nerves of these animals and repeated the experiment, that the animals did not get a fever. This meant that their IL-1 must be transmitting a signal up the vagus nerve, and that the signal going up the vagus nerve was critically important to initiate a fever response, because it was known that the brain obviously can regulate the fever response. So thinking about this as someone classically trained in neurosurgery and neuroscience, I wondered if perhaps we had stumbled across a reflex that others hadn't found. Because we knew, dating all the way back to Ramon y Cajal, that nerves or reflexes operate by a sensory arc that transmits signals into the central nervous system and activates corresponding motor arcs that carry information back to the body to mediate the effect. So on the back of an envelope, we sketched out the possibility that maybe there were reflexes. And we knew that this was based on the idea that the vagus nerve reflexes control heart rate, blood pressure, respiratory rate, insulin secretion, and gastrointestinal function. And there's more. And this was very well known at the time. But what we could not find any evidence of in the literature was that this was actually a way that the nervous system could also control the immune system. We frankly thought somebody must have thought of this before, but we couldn't find evidence that it had been studied. So we sketched out a hypothesis to study the question that perhaps cytokines activating sensory signals in the afferent vagus nerve could be mediating fever and anorexia. And we knew this to be true. And we proposed, if that was the sensory arc of an inflammatory reflex, perhaps by putting the 1493 in the brain, we had bumped into or stimulated the motor arc of the reflex, and maybe signals traveling down the vagus nerve would inhibit inflammation. And this presented immediately an idea of electrically stimulating the vagus nerve to prove or disprove this hypothesis. And so that's what Ludmila Borovikova did when she was a postdoc in the lab. Ludmila electrically stimulated the vagus nerve in animals, rats in this case, that had been given endotoxin to stimulate TNF production. So sham-operated animals made significant quantities of TNF after receiving endotoxin. When she cut the vagus nerve, they made even more TNF. And when she electrically stimulated the vagus nerve, the electrical vagus nerve stimulation suppressed the production of TNF, as had the 1493 in the brain. And when she measured the blood pressure in these animals, because TNF is a significant cause of blood pressure decreases during endotoxemia, when she cut the vagus nerve, the elevated TNF levels caused the animals to go into shock more quickly. And when she stimulated the vagus nerve of these animals with an electrode, she prevented the release of TNF and prevented the onset of shock. So this allowed us to formulate the hypothesis of the inflammatory reflex as a way in which the brain and central nervous system can reflexively control inflammation. Now, it was known then and before then that inflammation induces humoral anti-inflammatory responses, including glucocorticoids, IL-10, molecules like spermine, and hormones like MSH. And all of these contribute to counter-regulate inflammation. But this is a humoral system, meaning it's slow, it diffuses through the bloodstream, and therefore, if glucocorticoids were to immunosuppress or attenuate inflammation in the elbow, then because they would be traveling in the blood, the glucocorticoids would also exert immunosuppression in the liver, which may or may not be exactly necessary. On the other hand, we proposed a neural network that could control inflammation, could be much faster because nerves signal so much more quickly than hormones. They could be quite localized because of the anatomy of neural fibers and their association with specific cells. They could have shorter lasting effects, and specific fibers releasing specific agents could either inhibit inflammation, as we've shown with acetylcholine inhibiting inflammation through alpha-7 nicotinic acetylcholine receptors, or stimulate inflammation through peptinergic substances like substance P, which can stimulate inflammatory responses, or adrenergic neurons, which can either stimulate or inhibit. And so the idea of the inflammatory reflex was very interesting to us at the time, but we really needed to know a great deal more about its mechanisms. And so that's really what my colleagues and I have done for the last 20 years, is study these mechanisms in the hopes of making devices that could help patients. So a number of surprises popped up along the way. The first was when we looked in the spleen and found that the splenic neurons are adrenergic, not cholinergic. And so it turns out that this neuron in the spleen originates in a ganglia outside the spleen, and that ganglia is actually innervated by the vagus nerve. What happens is the vagus nerve signals terminate in a ganglia called the celiac superior mesenteric ganglion complex, and the adrenergic neurons in that ganglion travel into the spleen to release norepinephrine, which regulates these T cells, which are green because they express choline acetyltransferase, which is the rate-limiting enzyme for the biosynthesis of acetylcholine. So these are T cells that make acetylcholine. And the acetylcholine, once released, is the off switch. It interacts with alpha-7 to turn off cytokine productions by splenic monocytes and macrophages. But these T cells are only about 1 to 3% of the T cell population in the spleen and in the bloodstream. And these T cells appear to be identifiable and restricted to the expression of choline acetyltransferase. So this very small subset of T cells has a very important role in mediating the signals from the vagus nerve to the innate immune system, because if you deplete mice, if you deplete these T cells from mice, vagus nerve stimulation no longer controls or regulates the inflammatory capacity or the ability of those cells to produce cytokines. The alpha-7 receptor is required for the response to acetylcholine to regulate cytokines, and this is work done by Hong Wang when she was a postdoc in the lab. You can see here that wild-type mice receiving vagus nerve stimulation inhibits TNF, but the alpha-7 knockout mice, which are identical to these animals except they fail to express the alpha-7 nicotinic acetylcholine receptor, they fail to respond to vagus nerve stimulation, and they continue to make TNF in response to endotoxemia. So both these chat T cells and these alpha-7 receptors on monocytes and macrophages are critically important to the pathway. So those are some of the interesting findings we made along the way, my colleagues and I, and they can be summarized here. The electrical signals in the vagus nerve activate the splenic nerve. The splenic nerve releases norepinephrine, which interacts with choline acetyltransferase positive T cells. Those T cells, in turn, begin making acetylcholine, and the acetylcholine interacts with the alpha-7 receptor expressed by red pulp and marginal zone monocytes and macrophages, and that's what shuts off the TNF production. And it turns out that most of the TNF in mice and rats that arrive in the bloodstream during endotoxemia is made in the spleen. So that explains how this particular reflex can control TNF in this context. So all along, new questions arise. And the next question that really comes up is, well, the vagus nerve is a cranial nerve, and it arises in the brainstem. And we estimate at this point in the story that about several thousand fibers of the 100,000 fibers in the vagus nerve are sufficient to regulate the cytokine producing cells in the spleen. So the question we had next is, what is the nature of the cells in the brainstem that control the fibers that transmit to the ganglia that control inflammation in the spleen? So to address this question, Adam Krasel, a grad student in the lab, now a surgeon at Northwell Health here in New York, and Sangeeta Sivan, Professor Sivan, my colleague in the lab, Adam and Sangeeta attacked this problem by developing mice using optogenetics. Now, in these particular mice, the channel rhodopsin is expressed under the transcriptional control of the control elements for choline acetyltransferase. And that means what you're looking at here in the brainstem, in the region of the dorsal motor nucleus of the brainstem of these mice, are neurons in the dorsal motor nucleus which are expressing the channel rhodopsin because they express choline acetyltransferase. This gave Adam the opportunity to do stereotactic surgery and shine a laser light to activate those optogenetic neurons in the dorsal motor nucleus of the murine brainstem, and ask the question, what happens to the inflammatory reflex if I only activate the cholinergic neurons arising in the dorsal motor nucleus of the vagus? And what he found first was that when he put a recording cuff, a recording electrode on the splenic nerve and flashed the laser light on and recorded in the splenic nerve, that when the light was flashing in the animals that had been genetically engineered so that they were expressing channel rhodopsin on the cholinergic neurons, when the light is flashing in the brainstem, Adam's able to record pulses in the splenic nerve. And when the light is off, the nerve goes silent. And when the light is on, the nerve recordings are positive again. So to our knowledge, this is the first clear and direct proof that vagus nerve signaling, a parasympathetic, classic cholinergic parasympathetic nerve, regulates the signaling or the firing of a sympathetic splenic nerve or an adrenergic sympathetic nerve. This shows that the parasympathetic and the sympathetic nerve systems have an intersection that is functionally intact based on the cholinergic signals arising in the dorsal motor nucleus of the vagus, terminating in the celiac superior mesenteric ganglion plexus to control the origin of the splenic nerve. So we're also very interested in the nature of these signals and can they be tuned and specified to modulate specific cytokine responses. So up till now, for the entire first 30 minutes that I've been speaking here, I've been focusing on looking at cholinergic signals, controlling cytokine productions in the spleen in a way to inhibit inflammation. But we've also been looking closely with Eric Chang, another colleague and professor in the lab, with Sangeeta and with Teya Sava, at asking a specific question. If we modulate the frequency, if we modulate the pulse width, and if we modulate the duration of these electrical signals applied to the vagus nerve, can we find parameters that inhibit cytokine production, and can we find other parameters, electrical parameters, that stimulate cytokine production? And the simple answer is yes to both. So for instance, these are animals that have not received endotoxin, but depending on the nature of the electrical stimulus applied to the vagus nerve, there are conditions in which IL-6 production, for instance, can be enhanced, or IL-5 production can be inhibited. There's even conditions in which TNF can be inhibited in the absence of giving the animal endotoxin, and CXCL1 can be significantly stimulated. So this raises a very this raises a very large and important question, whether it's possible to develop electronic mechanisms to target specific neural subsets, to identify neural subsets capable of stimulating and preferentially stimulating inflammation or stimulating immunity, and distinguish those from neural subsets or neural signaling paradigms that inhibit inflammation or inhibit immunity. So we're extremely excited about that. Now, along the way in these scientific projects, I have had the distinct pleasure of co-authoring publications with almost 1,000 colleagues, and all of their names appear here. And I cannot express enough the importance of crossing fields such as immunology and neuroscience and physiology, crossing fields and collaborating, not competing. And I can think of no better venue than things like this in the middle of a pandemic, having virtual seminars to share complicated ideas. But nothing beats working together, and I really can't wait to getting us all back together again very soon, because there's still so much more to do. So for instance, we're in the middle of clinical trials now, embedding devices the size of your little fingernail on the vagus nerves of humans with severe inflammatory conditions in the hopes of treating diseases like rheumatoid arthritis and Crohn's disease. Now, my colleagues at Set Point Medical in Valencia, California, are currently in the midst of a breakthrough designated FDA-directed clinical trial in the US for rheumatoid arthritis. But there's already been several successful clinical trials, smaller trials in different centers that have been completed in Europe. The first of these was led by Paul Peter Tack and Frieda Koopman from Holland, as well as Ralph Zitnik and Yakov Levine at Set Point Medical, and Sangeeta Shivan here at the Feinstein Institute. And the first question we asked was, does the inflammatory reflex exist in humans, and is it functional? And to do this, Ash Metna, neurosurgeon here at Northwell in New York City, implanted patients with vagus nerve stimulators, and we harvested the blood of those patients and then harvested the monocytes in a whole blood system, stimulated the monocytes by adding endotoxin to the whole blood organ culture. And so what you see here is the amount of TNF being made by whole blood from patients prior to their anesthesia, after their anesthesia, but before the vagus nerve stimulator was turned on, and then 4 hours after the vagus nerve stimulator was turned on. There was a highly significant inhibition of whole blood or monocyte TNF production in these volunteers, and this was the first proof in humans that the inflammatory reflex controls TNF production and could be stimulated using a vagus nerve stimulating surgical implant. The same concept was then applied to patients in Europe. These were patients in Bosnia, Croatia, and Amsterdam. And these patients had severe rheumatoid arthritis. There were two cohorts of patients. Cohort 1 had failed drugs such as methotrexate and glucocorticoids. And cohort 2 had also failed to have any therapeutic benefit from biologics such as anti-TNF. And what you can see is that the patients were implanted and received, at the time of the surgical implant, they received a diagnostic stimulation. The diagnostic stimulation looks like it's having a small effect. The diagnostic stimulation is turning on the device in the operating room to make sure the impedance is acceptable and the device is functional. But the official trial treatment began on day 0. The device was turned on, and both groups of patients, cohort 1 and cohort 2, both had a highly significant treatment effect. This is looking at the disease activity score, 28 CRP, which means joint counts and serum CRP levels. This is a standard clinical assessment for rheumatoid arthritis trials. This is a very significant and meaningful clinical benefit. The curve in the middle, of course, is the combined cohort. This is a very significant response. The device was then turned off by design. And as you can see, soon after turning the device off, the patients began to get worse. The good news is when the device was turned back on again, the patients began to improve again. And I'm not going to show the data today, but it's been published. 1 year later, the patients continue to improve and they get almost back down to their treatment effect from the original treatment. So vagus nerve stimulation has a highly significant effect in treating rheumatoid arthritis patients. I flew to Mostar, Bosnia 10 years ago next week to meet the first patient. That's him, that's his physician, that's the chairman of medicine, the department chairman, her boss, and this is the electrophysiology technician in Mostar, Bosnia at the hospital. That's me looking a lot younger and very happy. Some people say I actually look happier than the patient, but trust me, the patient was extremely happy. He had spent several years bedridden in pain from rheumatoid arthritis. And at the time I met him, and as through recently, he is now taking no medications. He's back at work, and he's active with his children and playing sports such as tennis, being treated with a vagus nerve stimulator implanted in his neck for rheumatoid arthritis. So I told you about Kelly Owens, and I told you I got to know her. I told you she was suffering from severe Crohn's disease. And a few years back, out of the blue, I received an email from Kelly Owens. She was, as I said, very, very sick, but became aware of the clinical trials for this electrical stimulation of the inflammatory reflex that were going on in Europe. She and her husband, Sean, sold her worldly belongings and moved to Amsterdam and enrolled in the trial. And afterwards, she sent me an email that has the subject line shown here, which says, simply, "Thank you for saving my life." I answered the email and invited her to come so I could meet her. And she did come, and I have met her, and here she is today. This slide is out of date. She's now almost 4 years status post vagus nerve stimulator therapy. She takes no drugs. And I know that she looks like this because she works around the corner from me down the hall, where she is a manager and a director for patient outreach, because we get so many requests from patients and families to participate in clinical trials and offering to help, that Kelly now is responsible for directing the communication and the coordination with these patients, and in collecting a database of almost 1,500 patients who we hope to help with clinical trials someday. And that's her cane in the corner of my office that she gave to me as a gift because she said she doesn't need it anymore. So that's why we do this. The opportunity is enormous. The science is fascinating. The mechanisms are important, but the patients and their needs for understanding alternatives are what drive all of us. Where are we? I think we're at the very beginning. I don't think this is the end of the story by any means. I think we're at the very beginning of one of the most exciting times in the history of science. For those of you specifically interested in neuroimmunology, it's a growth industry. I made a comment before that when I graduated medical school, That was 1983. There were 11 publications that on PubMed that popped up under neuroimmune. You can do this. This is updated as of this morning. But as of 2021, that number is up to 3,491. The science of neuroimmunology is undergoing explosive growth, and I hope that translates soon into new therapies for patients who desperately need them. We're particularly interested in decoding some of the electrical signals these nerves are using to transmit these regulatory pathways. So Theo Zanos, an assistant professor here at the Feinstein Institute, with Sangeeta and Chad Boughton and others, we recently asked the simple question: if the vagus nerve is an electrical conduit that's transmitting information from the body's organs to the brain, and that information is being transmitted as electrical signals, essentially, compound action potentials transmitted in clusters of neurons, might it be possible to put a cuff, a recording electrode, on the vagus nerve of a mouse and decode nerve fibers transmitting specific signals? So Theo developed a naive Bayesian algorithm to isolate and decode these neural signals that were recorded with a cuff electrode on the surface of the mouse vagus nerve. Now, it's fascinating because here, mice were injected. What you're looking at are these are compound action potentials recorded through the cuff. And you see three different groups. This is in a mouse that was injected first with IL-1 and then later with TNF. Now, what you notice first is that after the IL-1 injection, there's a group of neurons that are orange here that are firing following shortly after the IL-1 administration. Now, this analysis was done in the time domain. So the simplest way to think of this are these are neural fibers that are firing at the same time after IL-1. And a different group of fibers are firing at the same time after the mouse was injected with TNF. But look at this. There's another group that just fires along and doesn't really seem to change its firing pattern regardless of the TNF or the IL-1. So this is a huge opportunity, we think, and we're at the beginning of this for decoding the electronic information transmitted in the nerves to garner information about the regulation of immunity. The other thing we're fascinated by is the fact that nerves can also stimulate inflammation. So one mediator that we've been interested in for many years, HMGB1, which my colleague Haichao Wang showed some years ago, is a pro-inflammatory mediator, but it's very special because it's found in the nucleus of all cells. It's found in the nucleus of all cells, but activated immune cells, monocytes, macrophages, can secrete HMGB1 as a cytokine by translocating it from the nucleus to the cytoplasm and then secreting it in exosomes. And when HMGB1 is secreted as a cytokine, it exerts a range of biological effects in inflammation that range from fever and anorexia all the way up to organ failure and death. Now, targeting HMGB1 with antibodies is a way to block inflammation. There are hundreds and hundreds of publications addressing the pathogenic role of HMGB1 in the mediation of inflammation, ranging from shock, endotoxic shock, bacterial sepsis, and even experimental arthritis in rodents. So we recently became aware that HMGB1 could also be released by neurons. And this prompted my colleague, Huan Yang, and her colleagues with Valentin Pavlov and Eric and Sangeeta and I to ask the question, might it be possible that neurons secrete HMGB1 and the neurons can stimulate inflammation? And so here what you're looking at are mice which were genetically engineered to express channel rhodopsin in nociceptors. And what you see is that when DRG neurons are exposed to 470 nanometer light in vitro, the HMG translocates from the nucleus of the cell to the cytoplasm of these neurons. And once in the cytoplasm, Juan went on to show that the HMG is released from these neurons. And this appears to be independent of cell death, because we didn't find evidence of significant LDH release. Now, the functional implications of this are profound, because when you subject mice to sciatic nerve injury, it causes inflammation distally in the paw and in the downstream tissues. So you can see evidence of that inflammation in wild-type mice subjected to sciatic nerve injury. TNF and other cytokines accumulate in the paw, and the paw becomes exquisitely sensitive to pain and pressure, with diminished threshold responses to von Frey filaments and other indicators. Now, HMGB1 floxed mice look like wild-type mice in both of these parameters, but when one depleted HMGB1 from the neurons, these animals now fail to develop inflammation in the paw, and they fail to develop the pain responses. This is really, really interesting to us, because it appears that the nerve injury creates the downstream inflammation. And this may, of course, be what happens in herniated disc or sciatica pain that complicates herniated lumbar discs, where the herniated disc puts pressure on the sciatic nerve or nerve roots, and those nerve roots now transmit HMGB1 release down into the tissues, and the patient feels pain in the calf, the thigh, or the foot, which is exactly what happens. What was really striking was a similar phenotype appears in the classic model of murine arthritis mediated by collagen antibody-induced arthritis. So here, this is the classic antibody response with arthritis developing in the paws of these animals subjected to collagen antibody injections with high levels of TNF. But depleting the HMG from the nociceptors treats the inflammation and reduces the inflammatory mediator production in the joints and paws of these animals. This is a very, very significant effect. So we're very, very excited. I think the opportunities for this are enormous. It's a tremendously fun and gratifying field to be working in. And I think that we hold in our sights, all of us interested in these problems, an idea that's actually quite significant. Because perhaps by understanding these neural mechanisms that regulate immunity and inflammation, and by learning other ones that we can only imagine today, maybe inflammation is curable. I'm going to say it. Maybe inflammation is curable, and maybe if inflammation was cured, lifespan would be prolonged. So I'll conclude there just by noting that we're always looking for good postdocs and grad students. Please reach out to us. We always have lab meetings that look just like this. We have a lot of fun, and thank you.
Thank you so much. That was a really, really nice talk. So I think I need to kind of mutate you and me so we don't have our micros on at the same time, because otherwise it's going to be a lot of echo. But I am going to start asking the questions that the audience ask here in the Q&A. So the first one is from Asia Rolls. Thank you so much for a fascinating talk and inspiring work. What would you consider will be the difference between neuronal reflexes and other types of neuronal control? When will you expect each of them to work?
Oh, hi, Asia. It's so nice to hear your question, and thank you again for inviting me to give this talk today. I think the sky is the limit for testable hypotheses to now look at specific aspects of neural control as they control specific aspects of immunity. So what do I mean by that? I think that the era we're in now and the tools we have from immunology and neuroscience are extraordinarily powerful. And I think we can build maps and based on neural and immunological mechanisms that we simply couldn't build before. And so I think to the extent that a question can be asked, then we can now generate a testable hypothesis. And that's what has me so excited about this. So in the case of your own, frankly, remarkable work, looking at the role of brain networks forming engrams in response to the presence of inflammation in the body, and the ability of stimulating those engrams to recapitulate the inflammation in the body of those animals. I mean, this tells us that specific neural networks correspond in real time to the status of specific biological endpoints, and that they're not separable. I mean, the power of your work and the power of the skills that we have today to ask specific mechanistic questions really boggles the mind, because what it says to me is that a physiological state in the body is linked almost instantaneously through neural signals, say traveling up the vagus nerve, into neural networks in the brain that are in turn linked to the physiological state of the body. And what your work says and our work says is that pushing the neurological state pushes the physiological state, and pushing the physiological state can push the neurological state. You can't separate them. And that frankly is I believe is a new way of thinking about how the immune system is controlled. It's a new way of thinking about sort of the physiology of immunology. And I think that that's the future. So yes, I agree with you. I am incredibly excited. And we're limited only by the number of specific hypotheses we can generate to form a functional map, the complexity of which boggles the mind, frankly.
Okay, second question is from Jeremy. Hi, Kevin. Amazing talk as usual. Sort of out of the blue question, but as immune responses are metabolically expensive, would you expect that the brain influences immune cell metabolism directly or indirectly? Applications in cancer, wound healing?
Hi, Jeremy. Thank you very much for your kind words. It's a pleasure to hear from you also. I think that the brain is agnostic to the specific receptor signaling pathways. What do I mean by that? I think that the brain networks are looking at the entire body as the receptor. So a specific macrophage, for instance, undergoing a Warburg response and making lots of cytokines is going to churn out lactate and TNF and IL-1. And those, arguably the lactate is a metabolic signal, but the brain sees the whole thing. The brain sees the specific effects of TNF and lactate on a specific neuron, but it also sees the indirect effects of the lactate traveling to another part of the body where it induces a different cell response, which is picked up by a different nociceptor that is also funneled up into that same brain network. Same for the TNF and IL-1 responses. The TNF is going to drop blood pressure, and the brain is going to register a whole set of inputs from those changing physiological responses. So I think the brain, if you call it the brain, the central nervous system is a series of oscillating neural networks or engrams, which is constantly changing in response to the physiology of the body. So do I think that the metabolic responses will be specifically controlled by specific neural signals that return in response to changes in the environment from the sensory input? Absolutely, yes, I do. And I think that we have the tools in hand now to begin systematically mapping those. In a funny way, so much of the important work that's happening now in neuroimmunology is building functional maps, specific neural circuits. No one circuit, no one engram, no one pathway is going to explain immunology or immunity. But there's going to be countless opportunities to study new circuits, new pathways, and new engrams linked to physiological states in immunity in the periphery. That's the opportunity for all of us, young and old, because there's not going to be a limit to the number of opportunities to produce this kind of information. And I believe some of them will have direct therapeutic implications for patients, because we can hack into these systems now. Building these devices is very, very feasible. You don't have to put them in the brain. You can hack in to these networks from lots of different transcutaneous and minimally invasive percutaneous strategies. So thank you for your question, Jeremy.
Yeah, it is really, really interesting. Okay, and I think I'm going to read the last question because we're a little bit behind time. So this is from Shane O'Mara. Inflammation is an evolved response, and as the old line goes, evolution is cleverer than you. With what precision? And when should you attempt to inhibit the inflammatory response? And what unexpected effects might occur? What a wonderful talk, by the way.
Yes, 100% agree with you. All of the evolution of the nervous system is based on obviously selection for adaptability to prolong the ability to pass on offspring. So that means that the evolutionary pressures on controlling inflammation, or any aspect, frankly, of immunity, the evolutionary pressures are not to immunosuppress or eradicate inflammation. And in fact, in the most optimal of laboratory conditions, using nerve stimulation, for instance, to block TNF, for instance, there's no way to block 100% of the TNF. So the vagus nerve signals, just to use an example, this is only just one example of many, but the vagus nerve signals in the nervous system only reduce the inflammatory response from the toxic, non-advantageous range to the safe, healthful, protective range. And in fact, that's how you'd expect evolution to work. So 100% agree with you. The opportunity we have to develop these maps and identify circuits that it may be possible to use for therapeutic intervention is grounded and rooted in evolution, as it has to be. That's where they came from.
Okay, thank you so much. So we are going to stop here, but again, I placed the Zoom link here in the chat, so anyone who wants can join. Thank you so much, Kevin, for such a really, really nice talk, and for everyone to join us today. So next week, we will have another seminar. Our next speaker is Colin Murphy, who is going to talk about transgenerational inheritance of pathogen avoidance, which is going to be also amazing. So I hope to see you all as well next week. And yeah, welcome to the discussion that we will have with Dr. Tracey now. Bye.